All right. Hello, everybody. Welcome to this is actually the penultimate talk in our 9th annual Women of Science event. This afternoon, we are very lucky to have with us Vicky Cerdeira, who is a student, a PhD student in biochemistry at Ucam. Her research is trying to understand the development of nervous systems in a very well-studied little worm called C. elegans. And she's studying how certain structures of neurons, such as dendritic spines, are formed and maintained within neural circuits. So she's going to be talking to us a little bit about that today. And I'm very excited to see and hear her presentation. I am going to be monitoring the chat, and I am going to be monitoring the participants as well. So for those of you who are online, you can feel free to enter questions in the chat at any point. And since I'll be monitoring those, I'll jump in and ask Vicky the questions. If you would like to ask yourself in person, you're more than welcome to turn your camera on and ask the question, or you can just raise your hand if you'd like, and I'll come to you when it's appropriate. I don't see Christy's class, Ashley, but I also don't know what name she would necessily be coming in under.
It did show up before. It said Christy's class, and then it disappeared because we hadn't let people in yet. So maybe she.
Oh, well, she'll come back. Yes, and in the classroom setting, if the students who are in the classroom, feel free to raise your hand. And the teachers who are there can call on you and interrupt as appropriate. Okay, so there's Lisi right there. Without further ado, I will hand it off to Vicky.
Awesome. Okay, so I'm just gonna put the sharing of the screen. I hope everybody sees my screen. I'm just gonna select my pointer.
There it is. We've got it. Yeah,
we got the screen.
Yeah.
Awesome. Okay, so hi everyone. Thank you for that awesome introduction. As Carl said, my name is actually Victoria, but I prefer Vicky. And I'm going to be talking to you about something super interesting, which is how can we simplify the study of the nervous system by using worms. But before starting with that, I'm just going to talk to you a little bit about myself. So this is a beautiful map that I found with the beautiful flags. And I just wanted to tell you that
That's lovely.
It's lovely, right?
Yeah, I like that a lot.
And I am from a country which is extremely down south compared to Canada. So I'm from Argentina. It was close to Brazil, Carl, so you were close enough. But I'm here, down here all the way from Argentina. And of course, I was a super cute little girl. Maybe I changed a lot, but well, at least when I was young, I was cute. And Argentina has this awesome amount of landscapes, which I think not a lot of countries have. And because of the tropical weather that we have in Argentina, we have up north, which is almost close to Brazil, we have really, really high temperatures, and we have these amazing waterfalls. At the same time, if you go all the way down south, so if you come all the way here, that is what it's called the southest province of all, because it's basically just neighboring Antarctica, and that's where we have our glaciers. We also have a lot of beautiful lakes and mountains. We have, honestly, I think Argentina, although I don't know a lot of other countries, but Argentina has one of the most beautiful landscapes throughout the whole country. And one of the biggest things in Argentina, if you ever go, is the food. And yes, I'm gonna make you hungry because it's the awesome food. And as you can see, we are a meat country. We love meat. We eat a lot of meat. We also eat, and you probably already heard or seen empanadas, which are usually filled with meat or chicken, or they can also be sweet sometimes. And we also have these awesome sweets, which are super difficult to find elsewhere. I would definitely recommend, if you ever go to Argentina, to at least try these things, because that's the basis of the country. And we're also one of the main producers of wine. Because of our different temperatures throughout the country, we can actually produce different kinds of wines and with different kind of grapes, and their tastes are completely different. So if you ever go to the SAQ, whenever you're in the legal age to drink, if you ever go to the SAQ and you see in the part that says Argentinian wines, then I can assure you, you're gonna like them. And so during my school years, of course, my passion was science in general, although physics was kind of problematic, a lot of math, but science in general, I really liked. I also worked a lot in languages. So of course, Spanish is my mother tongue. I did my whole secondary school in English, and I also learned French and Italian when I was in school. And I participated in the choirs and in all of the theater plays that we ever did at school. I was always there. History and geography were okay. They were okay, let's say, but math was my torture. Math was and still is my torture. So I think that I am the living proof that you can be a scientist without really liking math. If you have a calculator, then you're okay. And luckily, I also had the possibility of having extracurricular activities in school. So I would play handball. I also participated in a really, really nice international exchange in Ecuador, where I don't know if you're familiar with the International Baccalaureate exams, so the IB exams, but we were lucky that the international international exchange in Ecuador was all of the Latin American colleges that had the International Baccalaureate exam. We were reunited in Ecuador with the idea of generating activities that could potentiate the economy of certain towns that were underprivileged. And so I was super lucky to get to know a lot of these people from all over the Americas. We became really, really good friends. I also participated in a nonprofit organization, which was building houses for the underprivileged kids. So as you can see, the houses were made of wood. This is me right there. And that was a super cool experience. We actually built with this group that you can see here in the picture, we built 2 houses, one for a family of 4 and another in a family of 7 kids. And I also participated in the Red Cross, especially with Christmas events and those kind of more social events that we used to do. And so I finished school and I was certain about things that I wanted to do. I knew that genetics and the genome were super interesting. I was really intrigued about how some genetic information that we had in our cells, how can that extrapolate to an organ? How can the information in the genome generate these organs, these functional organs, and that if something is missing, then the organ is not functioning or there is a problem in the organ. At the same time, I was super interested in the cell. So all of the molecular mechanisms that the cell uses either to reproduce or to solve any problems that could come up, it was so intriguing to think that we know nothing, right? Especially at least at the cellular level. And I was super intrigued about how we could learn so much more. At the same time, I was super interested in medicine, but I knew that I couldn't be a doctor because I couldn't have a patient's life in my hands. So medicine I love, but I couldn't be a doctor. And at the same time, I knew that I wanted to follow research. And so I decided to study biotechnology and molecular genetics still in my home country in Argentina at the National University of Quilmes. And so during my college years, I was super lucky because my university had a more practical approach to all of the subjects. And so all of my subjects, of course, had a theory part, but then we could actually apply that theory into practical things. And so I got to see these awesome things. As you can see here, these are just a couple of examples, but this is, for example, an artery. So you can see these muscle cells that are there just surrounding this little kind of hole. And these little dots are your red blood cells. So yes, this is actually something that is happening in your body right now. And in this one here, this is a vein. As you can see, there is no muscle and the lumen is way smaller. And these are also the red blood cells here. And in this case, these are the bacteria that I worked in. I'm going to tell you a little bit about that later. And I put this picture because, well, it forms a little heart and it's beautiful. And so during my college years, the fact that I could immediately start doing practical experiences really confirmed that what I wanted was lab work and research work. And I joined the molecular microbiology lab in Argentina, where I started doing my thesis research. And I was lucky because I managed to present on a science meeting. As you can see here, this was my first poster. And this poster was selected for a 10-minute presentation, oral presentation. And I have to admit, it was super nerve-wracking because it was my first time presenting something as a poster, and I was selected as oral presentation. So I have to say I was really, really nervous, but everything turned out to be good. I got really good feedback. And with the information of my thesis, I actually published 2 science articles, and I am super happy to say that this one was accepted last Friday. So I am really happy that I could promote new knowledge with my thesis. And just to talk to you a little bit about this without going into a lot of details, as I said, Argentina is known for its winemaking industry. Now, the problem is that winemaking is a very expensive process. So I don't know if you know, but wine requires the production of ethanol or alcohol, and that is done by yeasts. But also there is a whole another step, which is fermenting and actually giving the wine the taste that it has. And for the wine to have that taste, we need a whole other type of bacteria that also pass through a fermentation, which is called a malolactic fermentation, but that doesn't matter. The name is way too fancy. So what I did was study different products that are food waste. So from the cheese industry, I used whey permeate, and from the cider industry, I used apple pomace. Those are things that just come out as wastes from the food industry. And I repurpose them to generate media that would be cheaper to generate the bacteria that then we can put into wine. So with these results, I showed that we could save up to 90% of the costs of winemaking if we could repurpose food wastes from this industry. And because this process was important and the whole project of the lab was important, we were actually cited in a newspaper because we are looking for using bacteria that is actually from the same region where you're taking the grape from. And that allows the wine to have this specific taste of a specific region. And for those of you who know wine, you know that every wine has its own taste. Every wine has its own leaving, like the tongue with a certain sensation. And that is because every grape has its characteristics and every bacteria has its characteristics. And so if we could manage to unify the bacteria and the grape from the same region, then that would give a specific flavor to the wine. So basically that was my thesis project. And that's where these 2 articles came from. But I also did other stuff outside of science, and I studied Argentinian Sign Language, where I had a 2-year course and I finished being an assistant in the communication of sign language. I also continued participating in the Argentinian Red Cross. I also joined ISEC, and I was also part of science animations for kids in birthday parties, which was basically taking science to fun and also explaining what happens. And I never forgot my dancing and my passion. So yes, I would go out every Friday and every Saturday and I would party. And then I would sleep my whole Sunday because of course that's what you do during college and it's okay. And so after all of my years of college, in July 2018, I finished my degree. I finished with my title of Biotechnology and Molecular Genetics with thesis in Applied Microbiology. And the reason why I'm putting this picture, and I know you might think this is weird, the reason why I'm putting this is because this is a tradition, and I think it's only Argentinian tradition. When you finish your degree, all of your friends or the people who love you and who come to see your defense of the thesis, they're gonna let's say, torture you a little bit once you finish your thesis. And so all of your clothes is gonna get cut up, so you're gonna be almost half naked. And then they're gonna start throwing you stuff. In best case scenario, it's gonna be foam and a little bit of color and, paints and stuff like that. In worst case scenario, they can throw, rotten food and stuff like that. It's a weird tradition, I know, but it's kind of like a rite of passage. It's really fun. You have things everywhere, but I think it's a nice rite of passage for kind of finishing a nice stage of your life, which takes a lot of effort and a lot of studying. So yeah, in July 2018, I finished my degree, and I decided, and as I finished my whole thing, as I told you, in Argentina, I did a couple of things, and I was lucky that with Claire, we managed to have a 2-month internship here in Montreal at Ucam, where she then offered me the position of PhD. Now, the thing is that because of my degree, I had to pass through 1 year of master for me to be able to do the fast track to PhD. And this was here at Ucam in Place des Arts in the Faculté des Sciences. And so why did I choose Ucam? So for all of you who don't know, and I'm guessing a lot of you know, but Ucam has a beautiful campus in the whole downtown Montreal. So we have here one in Place des Arts, which is Faculté des Sciences. Then we have at Berry-UQAM. Those, honestly, well, downtown Montreal is beautiful and Ucam is in the middle of it. So that was one of the reasons. The other reason was to improve my French, which I have to admit, it wasn't easy. But Ucam gave me courses in French and gave me French lab mates. And so I think I improved a lot of my French, and I hope to continue challenging myself because now I have to learn how to actually write French. I can speak it, now I have to write it. I'll get there, I hope I'll get there. And the third reason, of course, was Claire, who she's listening right now, an awesome PI, an awesome supervisor, and that is my third reason why I decided to choose Ucam. And so what do we do at Claire's lab? Well, we have three main lines. We have the nervous system, the study of the nervous system development, the nervous system maintenance, and the nervous system aging. I'm not going to go into all of them. I'm just going to be focusing on neuronal development and dendritic spines, which is basically my project. And as Carl said before, our research model is C. elegans, which is this beautiful little worm. And when I mean little is little, and I brought an example. So this is the plate. I don't know if you see it, but this is the plate we use to grow the worms. So for you to have a reference, this is my hand, okay? And inside of this plate, there must be thousands and thousands of worms. I don't think you're gonna see, but there's thousands and thousands of worms here. And this is what we use. This is how small the worm is, okay? Just for you to have as a reference. And so why do we use C. elegans? And so there are a lot of reasons why. One of them is because it's super easy to use. First of all, as I mentioned before, it's transparent. So it allows us to see this kind of magnificent stuff where you can label with different colors and you can see different things without having to even kill the worm. See, these are moving, that's in a plate, you just put it under the microscope and this is what you can see here. See, the pharynx is labeled in green, or then you also have some cells here that are labeled in green. Another of the things is that it feeds on bacteria, so it's super cheap to maintain and it's easy to produce that bacteria. At the same time, its cycle of life, it's pretty short. It's just for you to have an idea. Of course, it starts with the egg. Then it has 4 main larval stages, which we call L1 to L4. In L4, the worm is going to develop a vulva, and that is going to allow it to be a reproductive adult. Now, what is also interesting about the worm is that the reproductive adult is actually a hermaphrodite. What does a hermaphrodite mean? A hermaphrodite means that it has the female and the male organs in the same worm. So the worm as a whole, as an hermaphrodite, can produce its own progeny without needing the male. That is why the males are not very frequent in the worm. And the reason why males are still there, although they're technically necessary is because the males offer another genetic pool in case it's necessary to kind of move around some genes and change some genes, then the males are used for that. And one of the other things that is super interesting about the worm is that they actually can be frozen, and you can freeze them at -80 for months, -80 degrees, okay, for months. And then you just heat them up a little bit with your hand, you put them in the plate, and they're gonna grow again, and they're gonna generate babies, and they're gonna be super good. Other of the things that is interesting about the worm is that it has very powerful genetic. It's very easy to generate mutants from simple crosses or mutagenesis and generate rare mutant phenotypes that we can study to produce different types of knowledge. And this is something that we use a lot. And another of the things is that the worm has its genome fully annotated, which allows us to see that 70% of its genes have homologs with humans. So I'm not saying that we're worms. I'm just saying that part of the things that the worm has, its genes are so similar to us humans that we can study them with the idea of potentially extrapolating the information to humans. And because of its transparency, we can see this awesome things like neurons in colors, and we can see the whole body with all of the neurons labeled in a specific color. And that allows us to study the nervous system. And so the hermaphrodite has around 302 neurons. And because of the transparency and because of other mechanisms like using specific promoters of the genes, we can also label single neurons. So we don't need to do, for example, what researchers in mice do, that you have to take the brain, cut the brain, put a little slice on the brain, and then put in the microscope. We don't need to do that. We can just use the worm like that, a worm alive. You just grab one worm, you put it in the slide, and you look it under the microscope, and you would be able to see this kind of things. And because of the homology, as I mentioned before, then it has evolutionary conserved mechanisms. And regarding the dendritic spines, I'm going to go into that a little bit further. And so I would like to know, I don't think you think that, but do you think it's easy to study the nervous system? Do you think it's easy to study your brain? And I'm not seeing your faces, but I'm guessing most of you are saying, no, I don't think it's easy. And so just for you to have an idea, our brain, the connectome of our brain, has around 10 to 11 neurons and 10 to the 15 synapses. So that's 10 with a lot of zeros, as you can see in the picture. And it's virtually almost impossible to study that as a whole. Now, the benefit of C. elegans is that we reduced that, and C. elegans has 302 neurons and 7,000 synapses. And although it's still difficult to study everything together, it's not the same as studying 10 to the 11 neurons, right? I think 302 neurons is a number that at least it's more visually accessible, let's say. And so because of all of these reasons, then my project studies how is the development of the nervous system regulated. And so I study dendritic spines, and I also study the neurons in the simple C. elegans brain that can allow me to elucidate conserved molecular mechanisms to then hopefully extrapolate information about the human brain. And so the objective of my project is to study aspects of the nervous system development using Mao mutants in C. elegans. The concept of Mao, it's because it's maternally rescued. I'm not going to go into details. We're just going to call it Mao mutants. There are specific type of mutants. And my project has 3 main objectives, but I'm just going to be focusing in this conversation I'm just going to be focusing in the appearance or the phenotypic characterization. And for this, I'm going to show you 2 really interesting results that I have for behavior and for neuroanatomy. And so behavior, we studied, and we saw initially in the plates, as I showed you in this kind of plates under the microscope, we saw that the worms were moving not normally. They weren't moving as the normal worms. And so instead of studying them directly in the plates, Miriam, my student, she picked single worms and she put them in this drop of liquid, which is called M9. It has a mixture of salts. So she put the worm in a drop of water, and then she recorded their movement for around 90 seconds. This is what is called a thrashing behavioral essay. Thrashing because the concept of thrash is when the worm is managing to swim in that little drop of water. And so as you can see, this is a beautiful well-type worm. This is what we called a normal thrash. This is how the normal worm is supposed to look like. It's supposed to swim like in water or in a liquid. And so we decided to establish a coding color, more of a trying to quantify this movement. And we decided to establish a color. So green is gonna be normal, okay? Green is gonna be a normal thrashing. And so here you can see 8 different worms throughout those 90 seconds of recording. Now, our mutants, as you can see, the movement is clearly different. And this second video, believe me, it's not paused. It's just that the worm is paralyzed. Eventually, you're gonna see how it starts moving the little head and the little tail sometimes. And in this one, you can clearly see that the worm is kind of spasming, or it has kind of some ticks in the middle of trying to swim. And of course, the table based on the colors is completely different. You can see that there's mostly not green, and there is a lot of yellow and even red. So yellow is what we call spasms, as I told you, the worm kind of twitching. And then the red is complete paralysis. You can see that this mutant is basically paralyzed throughout the whole 90 seconds. Now, what is interesting about these results is that not only is each worm different between the worms, but what is interesting is that the same worm changes throughout those 90 seconds. And so this is interesting because that means that there is something that is being regulated throughout those 90 seconds in only one worm, okay? And that is why we say that the malmutants have an individual variability over time, because of course, every worm is different, but the same worm changes throughout those 90 seconds that we're studying it. And we also see the presence of spasms and paralysis, which could also help explain whenever we have more information, what is this about? And now the other aspect that I wanted to talk to you about was neuroanatomy. But before going into that, I'm just going to explain something because this might be difficult to see. So we have 2 different orientations of the worm to study it. So I know this is going to be complicated, so I'm just going to grab a pen so that it's easier. So if this is the worm, of course, a worm is a 3D object, right? So the worm is going to have the vulva as a reference point, okay? This is going to be the head and this is going to be the tail. And so if the worm is, as you can see here in the picture, if the worm is laying down in a way where you can see the vulva from either side, as you can see here, the vulva is on its side, then that's what we call a lateral view. And this is important because this is what I use to show you the results that I'm going to show you. So you're going to see this schematic, and I'm going to be emphasizing on the fact that it's a lateral view. And so this is the head, this is the tail, and this is the vulva. Okay, so the vulva is always going to be on its side. And I'm going to go into the colors later. And the other orientation is what we call the ventral dorsal side. Now the ventral dorsal, again, this is the head, this is the tail, but now the vulva is squished. So instead of seeing the left side of the worm, you're seeing the worm on its belly, either on its belly or on its back. And so what you're seeing is the ventral or the dorsal part of the worm. Now, why ventral or dorsal? The ventral nerve cord is as if I would tell you our medulla, okay? So the ventral nerve cord is where all of the neurons come from. And so the ventral nerve cord is the place where all of the somas of the neurons are gonna be there, and they're gonna extend their axon towards the dorsal nerve cord, okay? That's why in this beautiful picture, you can see, so this is the orientation, this side is the head, and this side is the tail. This little line in the middle is the ventral nerve cord. And these little bubbles are the somas of the neurons. And these little lines are the commissures or the axons of the neurons. So this is not what I'm going to use, but I just wanted to tell you that there are two ways of looking at the worms under the microscope. And so coming back to the lateral view, a normal worm has specific amount of axons, a specific amount of neurons established in a specific place. In this case, I'm just going to be focusing in two types of neurons, two groups of neurons. It's going to be the GABAergic neurons and the cholinergic neurons. And for the sake of, as you can see here, and I'm just going to go back, the GABAergic neurons, we're going to be seeing them in red, which are here. You can see them here. And the cholinergic neurons, we're going to be seeing them in green. Now, in a normal worm, on the left side of the worm, so again, lateral view, if the vulva is on this side, you're going to have a right side or a left side. In this case, I'm going to be focusing on the left side. And so on the left side of a normal worm, you're only going to have one commissure of the GABAergic red neuron. And you're also going to have 7 cholinergic, 4 before the vulva, so this is the vulva, and 3 after the vulva. Now, my malmutants showed these kind of things. They showed two main defects. The midline guidance defects, where we could see more than one commissure of the GABAergic neurons, and we could see more than 7 cholinergic throughout the body. And we could also see this lateral guidance defect, where they were neurons as if they didn't know where they were going, and they would just walk around the body without any final target. And I just wanted to put some pictures here for you to see how they look. So of course, this is not a normal axon. You can see that it doesn't really know where it's going compared to the other ones here that you can see them. So again, you see these little dots represent ventral nerve cord. So see, it leaves the ventral, goes to dorsal, but this one is clearly not doing that. And it's the same case here. This is GABA. So GABA in red. And you can see that this one it was apparently trying to reach, but it didn't. And then it had this misguided axon that didn't know where it was going. And so this is very interesting because then the malmutants show neuronal guidance defects, because it seems like the neurons do not know where they're supposed to be going. And so there's something that is missing. It's something that is not there. And so until now, I showed you that the malmutants have locomotion defects. And I also showed you that the malmutants have misguided motor neurons. And we decided to choose the DD neurons, which is a specific type of motor neuron, which is a GABAergic motor neuron, because it has the presence of dendritic spines. Now, what are dendritic spines? Well, dendritic spines were discovered by Ramon and Cajal in Purkinje cells, so that's a specific type of cell in the brain. And as you can see here in the picture, so this is a neuron, and you can see this little mushroom shape-like thingies that go out of the dendrite. Those are dendritic spines. And dendritic spines are known for incrementing or increasing the surface area of contact between neurons. And that allows them to receive axonal inputs and eventually transmit the information and have more contact between axons and dendrites. Now, the other interesting thing about dendritic spines is that they have extensive plasticity and dynamism during development, because you're going to see them grow, mature, and then go back to being immature and thin and small. And then they go back to being mature and mushroom-like, and then they go back to being like really nothing. So it is thought that that dynamic mechanism allows neurons to have nice communication and interactions and transmitting information. Now, of course, since the discovery of dendritic spines, a lot of the studies were done. And of course, your brain is full of dendritic spines. You have this in your brain in a lot of cells. And the interesting thing is that C. elegans has a lot of similarities to us mammals regarding dendritic spines. So C. elegans dendritic spines, their structure is similar. They also localize specific organelles. And they also show to respond to activity signals, that they can actually modulate that plasticity of spines because of the activity or non-activity that they receive. And so, as I mentioned to you before, our brain, the mammalian brain, around 90% of excitatory synapses occur in spines. And because of that structural plasticity, it is believed that they are linked to learning and memory. The benefit of C. elegans is that in particular in DD neurons, they also show these dendritic spines. And because of all of the benefits that I already told you about in C. elegans allows us to have an in vivo system that can allow us to study dendritic spines in real time in the worm without having to kill the worm. We can actually study this in a live worm. And that allows us to study how dendritic spines develop, how they mature, how they change throughout time. And it's very different to what we could actually do in humans or in mice, where you would have to kill the mice, take the brain, cut the brain. And so that gives you a specific time point. But in C. elegans, it would actually allow us to really study the development and the plasticity. And so this is, again, the cycle of life of C. elegans. I just wanted to show you that the dendritic spines start developing in the L2 stage. So here you can see, I don't know if you're going to be able to see it, but here you see this little sticky there in the black and white picture. You can see that that's the start of the development of a dendritic spine. And they continue developing all the way to the L4 stage, where they form, and you can clearly see the mushroom shape dendritic spine here in the black and white picture, and here also in red and green. And so we have a way of studying the development of dendritic spines using C. elegans. And in my case, in our MAO mutants, and this was the work done by Devin Oliver. She was a student from Claire, but at UMass, so in United States, and she collaborates with us. We found that, as you can see here on the wild type, so this is the neuron that we use to study dendritic spines, and you can see those little dots. Those little dots are dendritic spines. You can kind of see the mushroom shape. But in the MAO mutant, it's clear that you cannot see a lot of dots. So there isn't a lot of dendritic spines in comparison to the wild type. And you can also see that the dendritic spines are kind of clumped instead of being nicely distributed as in wild type. And so this is super preliminary. I don't have much more information than this. I have to work on, I'm gonna start working on it soon. But it's interesting that we have mutants, so our MAO mutants, that not only do they show a misguided neuron, but they also show defects on these little structures called dendritic spines. And so as a summary of my project, I start studying the animal and its behavior. How are the mutants affected phenotypically? What do they look like? And that is what we studied with the thrashing assay. How do they swim? Why are they swimming not normally? Then I also study the nervous system, because I study how are motor neurons affected in the MAO mutants. How are GABAergic neurons and cholinergic neurons affected in the MAO mutants? And then I go even deeper in the neuron and I study the neuron and the dendritic spines. And one thing that I didn't tell you about, because it was going to be too much information, was that I'm also going to be focusing in how are specific molecules, such as protein, enzymes, and mRNA, actually implicated in the development of dendritic spines. So this is a very big project. I didn't want to feed a lot of information, but this is basically the idea of my project. Now, of course, the question is, then why is my project important? And so, of course, first of all, science is curiosity and knowledge. I think all of us, we all always have questions, and that's the important part of science. And so my project helps to hopefully respond questions like, how do neurons develop? How do we function? How do neurons know where to go? Why do neurons go left instead of going right? And there are a lot of questions there, and that's the intriguing thing of the project. But apart from all of that, there are also medical applications to my project. And I'm going to talk to you about specifically two. So the first one is the impact on early onset schizophrenia. So schizophrenia, maybe most of you know what it is, but the particularity of early onset schizophrenia is that it appears before 13 years old. And this is a disease that is not very common. That's why it's classified as an orphan disease, because its prevalence is lower than 0.3%. But it is studied that this kind of schizophrenia, its main alterations are in dendritic spines because they show reduced density and reduced plasticity. They're not as dynamic as they should be. And so hopefully with my study, or my project, we could actually explain what are the molecular mechanisms that regulate dendritic spines, and hopefully that could explain the cause or the pathology of early onset schizophrenia. And the second disease is the autism disorders, because also this is a very broad aspect. Of course, there are a lot of causes for autism, but one of the main causes of autism, at least until for now, has been also alterations in the dendritic spines. So variations in size and number, again, coming back to those changes in morphology, not being very dynamic, not having enough dendritic spines to generate that contact between neurons, seems to be super important for the development of autism. So again, we hope that this project is going to allow us to expand knowledge in this and hopefully understand a little bit more and hopefully find potential solutions. And so with that, first I want to thank funding, because I don't know if you know, but you will know that science needs money. And without money, we can't do anything. And so first, I just want to thank La Faculté des Sciences and Sermon, because, well, it's Ucam and they have received me, and it's where Claire's lab is. They have also given me numerous scholarships that have helped me as an international student. I also applied for the master and doctoral scholarship of FAQNT, and I got both. So I'm super thankful for that. Of course, we have a lot of grants from other funding agencies, which of course I have to thank, because without them, we wouldn't do anything. I also want to thank the lab, which is, I couldn't have asked for a better lab. This is actually the most new picture. This is the updated picture. These are our three new students. Claire here showing a little plate to a little girl that was interested in the plate with worms. This was in the Montreal Science Museum in Women of Science, but in 2019. So of course, I am extremely grateful for being here in the lab. Claire is an awesome PI, and she's super intelligent, but above all, she's a great person. And so I'm really thankful for being here and for, being in her lab. And just to finish, I just wanted to talk to you a little bit about Ucam, because I know that potentially you're going to be applying either to baccalaureate or to master. And so we have a lot of domains of study at Faculté des Sciences. Right now, I'm in the Department of Biochemie and Biochemistry, but you have a lot of other different areas that are super interesting. There's going to be an open doors this Saturday, which I know is short notice, but if you're interested, there's going to be an open doors. And I'm leaving you here 2 important mails in case you're interested. And I'm also leaving you my mail. I'm open for questions about anything. So you can go ahead and send me a mail. I have no problem at all. And with that, thank you for your attention, and please go ahead and ask and interrupt.
All right. Thank you. Thank you very much, Vicky. That was a terrific presentation. Congratulations on the publication. Was that your first one?
My publications, well, actually, my first one was on February 2019.
Okay.
I was actually already here in Montreal, and I was finishing writing it up, and I got the news a month later that I got here to Montreal. And the last one, I received the acceptance on the 8th of October.
Awesome.
Of course, I worked well while I was here.
I guess so. Yeah. Congratulations. That's fantastic.
Yeah, thank you.
I'm also very glad that none of your close friends pelted you with rotten food. It seems when you graduated.
They did, but it was mixed with foam and colors, so I just had to deal with it.
That is an interesting tradition, I have to say. Does anybody have any questions for Vicky?
I can go back and forth with the presentation wherever you want.
Go ahead and speak. The mic is here.
All right. So question is, when the picture of the life cycle of the worm was shown, there was a certain thing called it started with a D, the Dower. What is?
Oh, the Dower stage. Oh, that's a good question. Okay, so the Dower stage, that's also one of the awesome benefits of the worm, is that if at the L1 stage there is no food, the worm can pass to this Dower stage, which is basically surviving to starvation until a food source comes up. And so the Dower is a worm, but it's just like a little bit thinner and smaller, because what it's just trying to do is survive starvation. Once the food is available again, then it passes directly to the L4 stage.
Oh, okay. So what's happening is like it takes longer, but it ends up going to the stage completely.
Well, actually, the Dower stage can take as long as there is no food. So at L1, the worm is super, super small, and it's barely, it's transparent. It's very difficult for you to see. If at that stage, you just take out the food and you leave it in a plate without food for months, it's gonna be in a Dower stage until there is food. If you pick that worm, Dower worm, and you put it in a plate with food, it's gonna take a couple of days, but then it's gonna get to the L4 stage. It's just gonna kind of revive after starvation.
So is it just that the gonads don't develop in the Dower? They wait until later? Is that
Yes, because L4, so the gonad is developing throughout the life stages, but L4 is when they start beginning to develop the vulva, which allows them to start developing oocytes. So the little eggs that start growing inside of the worm until a specific stage. And once they get to that specific stage, then the worm lays the egg and they continue developing outside of the uterus.
Any other questions? Okay, well, I have something for you. In the slide where you were showing the results of your measurements of worm behavior.
The thrashing assays.
Yeah, I mean, you were talking about variation. You were just noting that there's a lot of, but man, I find that to be a tremendous amount of variation. That the MAO9 mutant, I mean, there are ones that are completely, totally paralyzed, and there's others that barely seem to have any effect at all.
Yes, that's
kind of a ridiculous difference there.
Yes, that is super interesting. And actually, I didn't go into a lot of the molecular details of there, but we might have some results that the genes that are affected are related to molecular mechanisms that are super important for RNA. And so that makes sense with the development and how everything, of course, RNA is everything in the cells and everywhere. And so if we have genes that are actually affecting something in RNA, then of course, the worm is not going to be okay in a general state. Now, what is interesting indeed is what you're saying, the fact that some worms are completely paralyzed, but some worms don't seem as bad. So that is one of the main things that we're going to be focusing on indeed.
I have a question.
Okay,
Yeah.
sorry, I was trying to unmute myself and I hit the wrong button. Go ahead, Lisi.
Okay, so I was wondering how you make the in vivo images of the dendritic spines.
Ooh, that's interesting. I love your questions. Okay, so dendritic spines. The thing with dendritic spines is that they're super, 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 super small. And so we can't use a normal microscope. We have to use what it's called a confocal microscope. And then the confocal microscope allows us to have a sharper image and allows us to go really, really closer with a big objective that allows us to see really small things. Now, for us to be able to see dendritic spines, we need the neuron to be labeled. That's why I wrote it here, and I didn't go into details, but I can explain. All of the neurons that we label them with a specific promoter that is gonna drive the expression of our marker, which can be green, can be blue, can be red. In this case, FLIP13 is the promoter, and the promoter in this case is expressing the receptor of dendritic spine, the receptor in dendritic spines. And that is the thing that you're seeing in green. In the picture of the life stage here, you can see that there are two different things. You have a red and you have a green. Now, the red is labeling the neuron, the DD neuron. And the little green dots, as I mentioned before, they're going to be labeling the receptor. And so that is a way that you can localize the dendritic spine. There is where the dendritic spine is. Now, visualizing the dendritic spines, it's not as easy, and it's actually something that I have to optimize and that I have to be working on. So it's not as straightforward.
And I'm guessing they can only be observed in vivo?
Yes, well, in mice, they usually do it in sliced brain. And so that, of course, is something that is already dead. The benefit of C. elegans is that we can indeed see them in vivo. And so you could see them in different stages. You could also do a time lapse and have the worm and take the pictures of the worm at different stages and see how they develop. So yes, in the benefit of C. elegans is that you can indeed do it in vivo.
Okay, thank you.
Yeah, thank you.
So, forgive me if I'm wrong about this, because as you were going through presenting various steps of the scale of what you're looking at here, I believe I noticed that you're seeing in your mutants, you're seeing more neuronal projections of the two various kinds of neurons, but less dendritic spines. Is that correct?
Yes. So
how do those link together? Do you have an idea, a hypothesis, or is this well understood of how these things will link together?
Well, that's a very wide question. Right now, since we're trying to characterize as much as we can of what is happening with this mutant, what do we see? We're first trying to, characterize what do we see. Now, because of the information that we have that it might be related to RNA, then we are kind of generating the hypothesis that, of course, RNA, the mechanism of RNA is affecting in some way that it's affecting the whole worm. So it's affecting how the worm moves, it's affecting how the worm generates its axons and how they extend. One thing that I didn't mention, but because I didn't want to go into details, but our male mutants, they have 30% of embryonic lethality and 30% of larval lethality. And so not only is it affecting how they move, but their larvae are dying and the eggs are not developing. And at the same time, the neurons are misguided. And at the same time, the dendritic spines are not there, or they're bad placed, or they're not a lot. So of course, RNA is everywhere. And of course, if you mess up with RNA, then you're messing up with the whole system. Now, the question is why or how?
Yeah, that's always the question, isn't it?
Exactly.
For autism in humans, I don't know much about it. How well understood is this idea of dendritic spine issues? I'm throwing that out there. I'm not necessarily sure you're going to be able to answer it, but this was completely new to me. I was really interested.
Yeah, well, actually, there are a lot of neurological diseases that seem to start acquiring the idea of dendritic spines. And so, well, although I can give you a lot of specific information about autism because I haven't gone into a lot of details in human, but because of how axons and neurons modulate information, then the more surface you have of contact, the better information or transmission of information you're going to have. Now, because of dendritic spines are that the plasticity and the dynamic interactions between, oh, I'm going to put a dendritic spine to have this contact. Oh, I don't need it anymore. Then I'm just going to shrink in. Oh, I'm going to need it again. Then I go back again. If I don't have that, and then the dendritic spine is just stuck, then you're either going to have either too much transmission or not enough transmission. And so basically that's what they're suggesting. They're suggesting that if there's not enough dendritic spines or that they're not mobile or dynamic enough, then the information seems to be collapsing somewhere, and that might be the cause.
Wow, very, very interesting.
Yes.
Yeah, well, yeah, really, really interesting. I'd never even heard of that before, so thank you. I'm not seeing any other questions in the chat or no hands, I believe, from the participants. So, well, it's 3:30, perfect timing. Thank you so much, Vicky, for your time. We really, really appreciate it. That was a terrific presentation. And if you don't mind just sticking around for a couple of minutes, just to finish things up. And thank you to the classes and everybody online for participating. We hope you enjoyed this. There's one more talk tomorrow, and one little plug for our research methods course. We're throwing that out there. I forgot the first couple of ones of these things, but we have a research methods course. If you are in your third semester, or you're thinking towards when you're going to be in your third semester, we have a fourth semester course offered in the winter where we allow students, give students the opportunity to go take or go into a research lab in university and conduct some actual research there and get class credit for it. If you're interested in this, just send me a bio and I'll point you in the right direction. So one last little plug for that.
If you need any questions about research and research life, please, I already left you my mail. I'm up for any mail if you ever need.
And if you want it and you don't have it from this, contact me and I'll get it to you. Thank you again, Vicky, so much. Thank you to everybody. Have a great rest of the afternoon.